Exciting news! Adults can still grow new brain cells. For years, the potential for adults to generate new brain cells has stirred debate among scientists. But now, a remarkable study has cleared up the confusion, showing that the human brain can indeed produce new neurons well into adulthood. This discovery centers on a process called neurogenesis, which predominantly occurs in the hippocampus, a pivotal brain region crucial for learning, memory, and emotional regulation. Simply put, our research decisively confirms that adult human brains are capable of generating new neurons, shared Marta Petrolini, one of the lead authors of the study and a researcher at the Karolinska Institute, in an email to Live Science. Many experts are now endorsing this finding as a compelling argument for neurogenesis in adults. Although this single study doesn't provide absolute proof, it presents robust evidence supporting the notion that stem cells and neuron precursors are active and multiplying in the adult human brain, according to Dr. Rajiv Ratan, CEO of the Burke Neurological Institute at Weill Cornell Medicine. He explained, this serves as a remarkable foundation for the clinical neuroscience field, unleashing the power of cutting-edge science. The researchers utilized state-of-the-art methods, blending single-nucleus RNA sequencing and machine learning to meticulously analyze brain tissue samples sourced from various international biobanks. RNA indicates which genes are actively expressed inside cells, and machine learning is a form of artificial intelligence adept at analyzing large datasets. Since the 1960s, it has been established that certain animals, such as mice, rats, and some types of non-human primates, produce new brain cells throughout their lives, particularly in the dentate gyrus, a subregion of the hippocampus. However, accessing well-preserved adult human brain tissue has long been a significant hurdle. Obtaining human tissue generally involves autopsies or surgical samples. The way these samples are managed can conceivably obscure the presence of new cells, Paterlini explained. Leveraging modern technologies, the researchers succeeded in overcoming this issue. By analyzing over 400,000 hippocampal cell nuclei from 24 individuals and several techniques, in 10 additional brains with varied techniques, the team looked at samples from ages ranging from 0 to 78 years, including children, teenagers, and adults. Employing advanced imaging methods, they precisely mapped the locations of new cells, revealing precursor cells dividing next to fully developed neurons, area where animal research has confirmed adult stem cells are located. But here's where it gets controversial. The team didn't just observe these dividing precursor cells in young children, they also identify them in teenagers and adults. These include stem cells capable of self-renewal and giving rise to other brain cells. The hidden potential of adult neurogenesis. The latest technology permitted the researchers to spot new brain cells in diverse developmental stages and conduct studies that were previously beyond reach, Dr. Ratan noted. They also tagged the proliferating cells with fluorescent markers, creating a machine learning algorithm tailored to identifying neurogenic stem cells based on historical animal studies. This innovative approach made it easier to study brain cell formation in older individuals, Ratan added. As anticipated, children's brains produced a higher number of new cells compared to adolescents and adults. Interestingly, 9 out of 14 adult brains analyzed with one technique showed evidence of neurogenesis, and surprisingly, all 10 adult brains examined with a second method indicated new cells. The few samples displaying no new cells leave questions about the variation in adult neurogenesis. Looking forward, implications and future prospects. Considering future possibilities, researchers could investigate whether the observed neurogenesis in adults is a response to neurological conditions like Alzheimer's disease or a marker of overall brain health, suggested Dr. W. Taylor Kimberly, Chief of Neurocritical Care at Massachusetts General Brigham. He continued, identifying and understanding these cells' regulation can have significant implications for observing how they change over time and their connection to diseases, he mentioned. By comparing patients experiencing dementia to superagers, those who maintain cognitive resilience late into life, we might discover if a correlation exists between neurogenesis and disease. This could unlock new treatment opportunities. Though the specific therapeutic strategies for humans remain under exploration, Karolinska emphasized the mere fact that our adult brains are capable of generating new neurons changes our perspective on lifelong learning, recovery from injuries, and the incredible potential of neuroplasticity. Teresa Sullivan-Barger is a distinguished freelance journalist who specializes in health, science, and the environment. Her work has been published in prominent media outlets such as the New York Times, the Boston Globe, and Los Angeles Times, among others. Residing in central Connecticut, she is an advanced master gardener with a passion for wildlife-friendly gardening, especially for pollinators and songbirds.